First of all, I would like to apologize for giving this talk in English, but I believe you all speak better English than my German. So, so the talk I want to present to you today is biofilm activities at Empa. So here's a picture you see. It's bacteria cells on a polyester fiber. And before I start my talk, I want to come to the common understanding about the biofilm. So what is biofilm? Biofilm is defined as a microbial community and seated on a surface, material surface. So they produce also a matrix which contains polysaccharide, proteins or lipids, and extracellular DNAs. And all these polymers are defined as extracellular polymeric substance. So they form a very strong network, and they are very much resistant against antibiotic treatment. Once the biofilm is formed, and it's very difficult to remove them as well. And you see biofilm everywhere. Yeah, on nature material, like in stones, rocks, or hot springs, and also in our body. In the guts, we need bacteria for digestion, and also in our mouth. And you also find biofilm on artificial surfaces, like contact lens, toothbrush, and also on the water pipes. This is a big problem. And very interestingly for today, it's this biofilm formed on the medical device. They can cause a huge problem for health and hygiene. And in our lab, we are mainly interested in materials, medical-related materials, and we are interested in studying the interactions of the biofilm and bacteria on these medical devices, medical materials. So as you can imagine, when you have a medical device in place into the body, and immediately this device will be covered by a body fluid. For example, when you have a catheter, urinary catheter, then in place, then the catheter is immediately covered by urine. Or you have a dental implant, then the dental implant will be covered by saliva, or normal body implant will be covered by blood. So all this body fluid will form a conditioning film. So this film will promote bacteria adhesion. So here, the green color is the bacteria, illustrate bacteria. So they promote bacteria adhesion. And then bacteria will proliferate on this material surface and get maturated. So when they grow, the biofilm grow to a certain size, then the bacteria cell can be released from this biofilm and then find new locations to, again, attach on the surface. And you can imagine when you have different body locations, you will have different types of biofilm, like different bacteria species or different body environment. And also on top, you have this different environment and different materials. So what we are interested in is we want to establish an in vitro model which can mimic the in vivo conditions. And to do this, we need to have very well-equipped labs or infrastructure. Here, just example, we have this plate reader for static analysis, so meaning to analyze the biofilm formed under static conditions. And we also have different microscopies. Here is just an illustration for fluorescent microscopy. We also have a different flow chamber system. So we can grow the biofilm on a material surface and the flow conditions, which is more mimicking the in vivo situation. And we also have established in our lab different methods to assess, to evaluate biofilms. Like we can evaluate the EPS, this extracellular matrix. We can also evaluate the total biofilm content, or just bacteria cells, or even to differentiate the viable cells from dead cells. And next question you would ask, why do we need to establish such method? So as you have heard from Katerina's talk, indeed, in this biofilm field, there are many methods available, standardized methods, but these methods are not really suitable for biofilm tests. They are mainly used for this antimicrobial, antibacterial test, but not really to test biofilm, means the bacterial community on the surface. That's why we are also interested in set up such a method to really reliable evaluate biofilm found on the surface. And now I would like to give you some examples of the biofilm activities. And one of these is we want to remove a biofilm from medical device. So here you see this is endoscope. And endoscope, you have different opticals. These optics are very precious and expensive. And also, when you clean or reprocessing the endoscope, more than 90% of the endoscopes are still contain biofilm, means bacteria still attached on the surface. So this causes a potential contamination to the following patients who will need this endoscope. So one of the companies came to us and asked whether we can help them to develop a detergent which can remove the biofilm efficiently. And they have sought to use a mechanical method to remove the biofilm, but it's simply not possible because some of the channels, endoscope channels, are very, very tiny. It's almost not possible to reach there mechanically. And also the optics here on the surface, they are too pressure to be cleaned mechanically as well. And they also tried with chemical method, but again, here it's too harsh because you need a very high pH, like alkaline range, or very low pH. This will harm the optic. That's why they ask us to develop a green and effective method, which are friendly to the endoscope. So first, we have analyzed what is on this endoscope, so it means biofilm. So we have to know what kind of biofilm is formed there, so it means what kind of bacteria species, and what kind of composition of this EPS, means protein, DNA, and sugar, the composition. Based on this, we were thinking, how about enzyme? Because enzyme can cut these polymers very gently and mildly. And then with this thought, we test different enzymes and see how effective they can remove the biofilm formed on the surface. And here is our present you some of the data. So first, we formed the biofilm of Pseudomonas aeruginosa, and then tested and washed the biofilm, and then tested what is left after treatment with the detergent. So here, this is a control, means only wash the biofilm with water. And this purple-colored lines is the detergent available in the market. And this green line is what we developed for the company. And here you can see, this is another type of biofilm, very commonly found in the endoscope. It's Staphylococcus aureus. And we also did similar tests. Here you can see with the newly developed product, we can reduce the biofilm dramatically. So only afterwards, only around 10%, even 5% is left. And this is definitely not enough because we have to also get approval from the government. Then we have to do an ESO test. And for this, we did a dummy test with a real endoscope piece. And then here's the control, means washed. We formed the biofilm on the endoscope, and afterwards treated with either water or our detergent. And then here you see, this is green-colored spots. They are bacteria. They are biofilm on the endoscope. Here's many bacteria cells. And then after treatment with the detergent, you see very few cells left. This is a lot, because after this is just remove the biofilm. In the second step, you have to kill the bacteria, meaning you have to use a disinfectant. So we have done this experiment to see how many, first we calculate how many cells left over after treatment. Here you can see we can reduce cell number by log 2.5. And then we also afterwards we treated with the disinfected, we can kill more than 99.9% of the cells. And then we also measured, based on this ESO test, we have to measure how much protein left, how much sugar is left. So in both tests, we can, like protein only 5%, more than 95% protein are removed, and more than 90% of sugar are removed. So this is fit to the criteria of the standard request. And now the company is going to launch the product in the next 7 months. Another project is we want to help the company to develop non-biocidal and anti-adhesive textile coatings. And since due to the time limitation, I cannot go to in details. Here is just, and the company has developed, this is sanitized and Swiss test. They have developed a special coating which can prevent bacterial adhesion. And we have established the in vitro model to test whether this coating really anti-adhesive against bacterial adhesion. And here we reached more than 80%. With one of the coating, we could reach more than 80% bacterial adhesion. And based on this result, we got, in 2013, this Swiss Technology Award. And also based on this success, we have a continuation project with these two companies. And here, we not only want to reduce the bacterial adhesion, but we also want to reduce the malodor formation on the textiles. So this is just ongoing project. So another example of this biofilm example is we want to treat the endodontic infection with special formulations. Here, I just show you a healthy teeth. They have a structure like this. But when the teeth is infected, especially down to this endodontic root canal, and the biofilm can be formed. Here show you this biofilm formation. And the normal treatment is you have to open the teeth and clean the channel. Then afterwards, you have to fill the channel with this gutta percha and also the cement. Then afterwards, you have to fill the final filling and the crown, and hopefully you have the bone healing. But very often, after this normal treatment, and you don't have efficient removal of non-necrotic tissues. And also, you cannot access these tiny channels, tiny canal in the teeth. So it's very difficult to remove all the infections, means remove all the bacteria. So very often you have reinfection. So in this project, what we want to do, we want to have helping the company to develop cleaning agents to better remove the non-necrotic tissue, and more importantly, we want to destroy the bacteria in a sustainable way, not only during the treatment, but also in the following months or years to come. And this bacteria in the small channels can also be killed. So this is started just recently, ongoing project. And now I come to a more research topic we have. We want to study the surface topology versus bacterial adhesion. So here, this morning, you have seen many surface fabrications, like micro fabrication or nano fabrication. And in this project, we want to study the nanostructured surface, how they impact the bacterial adhesion. And this is structure fabricated at EPFL, and we were provided by this EPFL, by our partners with nanostructured gold surface. So these nanostructures are made of gold. So this is a silicone substrate, and here's a tungsten interface, and here they made gold nanostructures. And first, we just have a non-structured surface. Here you see the bacterial adhesion. These green colored spots, they are bacteria. And if they are alive, they are green. If they are dead, they will be red. So here you see some red spots, but very few. So you see there are living cells attached on the surface. And when you look at the SAM picture, and this is Staphylococcus aureus, so you see they are healthy cells. And then now we come to this nanostructured surface. We have structure around 100 to 400 nanometer height, and the distance should be around 20 to 40 nanometer in distance. And with the same sample, we see most of cells are red, meaning they are dead. They attach on the surface, but most of them are dead. Here you see some green color, means they are still alive, but the majority of the cells on this nanostructured surface are dead. When you come to close to have to look at the SAM structure, you see the cell somehow sits on a surface, gets dried out or gets dissolved. And you come even closer, you see this cell, this nanopillar, and the cell somehow strongly attached to this nanopillar. And it seems like some surface tension, and it's there, it somehow breaks influence the cell membrane, and then cells start to not happy, and then start to lyse. We don't have a clear explanation for this phenomenon. We just observe this phenomenon, and we are also happy if somebody has some ideas to give explanation. And yeah, now I come to the summary. So we are interested in development of in vitro biofilm models, which mimicking the in vitro conditions. And because this in vitro model will allow us reliable for better assessment of anti-biofilm coatings, like detergents, materials, or even surface. And also, ultimately, we want to develop this in vitro model for better prediction of the interactions between bacterial cells and materials. And this work is, most of the work are carried out with our partners, industry partners and academic partners. And also, this is our team in the biofilm group. Yeah, thank you for your attention.